So welcome everybody to today's ERCC webinar. I'm Roger Alexander, the Scientific Outreach Coordinator for the Extracellular RNA Communication Consortium. Today we have Gustavo Stolovitzky here. He's the chair of the Exploratory Life Sciences Program at IBM's TJ Watson Research Center in Yorktown Heights, New York. I asked him to come because we're setting up an exRNA data analysis workshop in April. You guys should take a look, exrna.org/workshop. And sort of one of the culminating things we want to do at the end of that workshop, after discussing different techniques people have used, open problems in the field, is to talk about exactly what Gustavo is going to talk about today. And he's the right person to do it. He's the co-founder of the Dream Challenges, which is a framework for setting up competitions for big data analysis in a variety of fields. And so Gustavo today is going to talk to us about how we can design a challenge around exRNA data. Looking forward to it, Gustavo.
Thank you, Roger. Thank you, Christine, and the organizers. It's a pleasure to discuss with you this concept of crowdsourcing and benchmarking. And so the outline of the talk is going to be how benchmarking competitions can be a path to reproducible research. Then I will talk about my efforts in this field, which is the founding of the Dream Challenges and what we have been doing in the last 13 years since we started. And we'll finish with a proposition or some ideas, not really a proposition, but some ideas as to how Dream Challenges can help benchmark the field of extracellular RNA transcriptomics. A field that is not completely foreign to me because in the last 5 years, I had a project on extracting exosomes from fluids and sequencing what is inside. So I run into the problems that many of you also are facing day in and day out. So let's start by discussing one topic in the literature that was very active at the end of 2019, beginning of 2020, which is benchmarking or actually developing, not benchmarking, but developing algorithms to detect cancer in screening mammograms. As you guys know, breast cancer is a terrible killer in women, and programs for screening have been very helpful. And the idea now is to see whether artificial intelligence or image analysis could do the job. And so there have been a number of papers reporting work, probably the most prominent of which appeared exactly a year ago, a little bit more, in Nature, from a group at Google. Now, if you look at it had a lot of press, and so if you look at the reaction of the community to this, it was a little bit not all was roses. There was this paper that appeared a little bit later that says criticizes the Google paper on analyzing digital mammograms because of lack of transparency and reproducibility. The criticism in the paper is that the absence of documented methods and computer code underlying the study effectively undermines its scientific value. And transparency in the form of actual computer code used to train a model and arrive at its final set of parameters is essential for research reproducibility. Also, there is an issue of how do we know that they didn't overfit? How do we know that they treated the data right? We only have their words in the paper, but that's it. So this motivated another paper about 3 months ago that says that AI, and in general, data science, I would say, is wrestling with a replication crisis. replication crisis that is not foreign to other areas of science, including experimental science. But this is a good paper that discusses this. And the author says this, it says, when we saw the paper from Google, actually this is not the author, but it's referring to someone saying, when we saw the paper from Google, we realized that it was yet another example of a very high-profile journal publishing a very exciting study that has nothing to do with science. It's more an advertisement for cool technology. We can't really do anything with it. So it's interesting, you know, this doesn't mean that what the Google team did was wrong. It's simply that it's unverifiable and as such doesn't belong in the enterprise of science as a reproducible and verifiable endeavor. We came a little bit later in March 2020 with a paper on work that we had done a few years before, and I will report on it later, in which we evaluated in a blind data set in ways that I will discuss more in detail, about 500,000 mammograms. We gave it for training, and we kept a test set to verify the training to see whether they generalizes. And we made some observations that are a little bit at odds with what the previous papers were saying. The previous papers were, they wanted to show that their algorithms were good. And so that you cannot be the judge, jury, and executioner of your own work. So what we, in a more dispassionate way, wanted to say is that we are not doing better than radiologists yet. Our artificial intelligence is not there yet. There is not only lost, there is a lot that we can do, but it was a more objective way of doing it and benchmarking all algorithms against each other on a common framework. Now, this is another example of something that went well, and some of you must know about this. There is a competition called CASP. CASP stands for Critical Assessment for Structure Prediction, that basically has been running since 1994, trying to find ways to benchmark algorithms that take amino acid sequences and predict the protein, the folding of the protein. And it runs every 2 years, and it has really catalyzed the state of the art in protein structure prediction. It also monitors the progress of the algorithm and establishes the state of the art of the field. CASP chooses protein structures that have been determined but are not yet published, and uses those as targets for team to predict something that is not yet published and not known in the public, but the sequence can be given to the predictors, and they can eventually say how well, the metrics to compare the prediction to the real X-ray crystallography can be done in order to determine how well an algorithm is performing and compare one algorithm with others. And a company, an artificial intelligence company that does deep learning called DeepMind, you may know about it from other work that they have done, such as, for example, playing and winning over the Go world champion, what is called AlphaGo. But they have been participating in CASP using algorithms to predict protein structure prediction. And they have, to some extent, made a definitive advancement to a 50-year-old grand challenge in biology, which is protein folding. On the right, you can see the predictions in blue and the real structure in green, I think it is. I'm colorblind. And so you can see that it's very close. It's indeed 1.6 angstroms on average, atom by atom, and 1.6 is about the error of X-ray crystallography. So you cannot do much better than this. And on the left, you see the evolution of the field and how they were kind of stuck at this GDT metric. 100 is perfect, 0 is random. And you go and you see that in 2006, 8 through 16, they were kind of stuck in a plateau. And when AlphaFold from DeepMind started to produce their predictions in 2018, already there was a leap. And in 2020, they are at 90% of this metric GDT. The point I'm trying to make is not, I don't have shares in DeepMind, but what I'm trying to say is that it is necessary to have an objective assessment of how they are doing and compare everybody against everybody, and to some extent keep doing these challenges to see how we are evolving. And very good. I saw a question. Let's ask questions later so that we have more time to discuss. Now, CASP is an effort from computational biologists and structural prediction biologists that is based on a process called crowdsourcing, which is something old. Even Bernoulli crowdsourced the problem of the brachistochrone in mathematics, and in 1714, the British Crown crowdsourced the problem of predicting the longitude at sea. But in its modern embodiment, the word crowdsourcing was coined in 2006, and what it means is a methodology that uses the voluntary help of large communities to help problems posed by an organization. And in the case of CASP, the organization is CASP, and the voluntary nature is sometimes not so in some tasks such as the mechanical Turk, but basically the idea is that you ask people, please solve this problem, and they feel a motivation to do it for different reasons. So basically, the organization has a problem, it crowdsources to a community of solvers, the solvers propose solutions, and the problem may be solved or not, or partially solved. When it comes to benchmarking, the problem typically has to have a known solution, at least in one instance. I mean, for example, we don't know exactly how to predict breast cancer from mammograms, but we can have a cohort for whom we know whether the women had or not. And in that instance, or that cohort we know. And so we can crowdsource the data, and the people can analyze the images and say, I know who has cancer, who doesn't, and propose the solutions that can be evaluated against the ground truth. Now, what do these people that are solving your problem gain? In general, they can answer a very important question if they are scientists, scientific researchers, is how accurate my algorithm is. Because usually it's very difficult for anybody to assess their own algorithms. Actually, there is a famous psychology paper that reports that 80% of people who were asked think that they are better drivers than the average. And we know that we cannot all be better than average, right? So the notion of self-benchmarking is important, but also these people have other incentives to participate, and we can discuss that in the questions. So what are the benefits of benchmarking by crowdsourcing? The first one, which I just mentioned, is performance evaluation. We can, by organizing a competition such as CASP, where AlphaFold participated, we can make an unbiased method assessment and discover the best methods amongst many, and also compare methods, even determine the solvability of a scientific question, or the level to which this question is now bringing best solutions that we had in the past. And it's important this is done in a blind situation, like double blindness in a clinical trial, for example, because otherwise there are conscious and unconscious biases that can creep in. And so we can run into what we have called the self-assessment trap. Now, there are other benefits of crowdsourcing. One is one other benefit is that if 100 teams work together or work independently on solving a problem, in a way you are trying 100 different ways of solving the problem. And if you find the best, then you have done, in the time that it took the solvers to try to solve the problem, you have done the work that one team would have done in 100 years, and you do that in 1 year because you have 100 teams. So it's basically an embarrassingly parallelizable problem. But there is more to it, because these people that you can get them together to solve a problem, eventually what we do in DREAM is we ask them to participate in what we call the community phase, in which the participants that previously competed, now they are collaborating. It's a collaboration phase. And so it is possible to foster collaborations in a community. And now those people that have been working individually to solve a problem, now they can share their experiences, learn from each other, because they have thought about the same problem on the same date, and they can figure out what went right and what went wrong. And in a way, also, we can aggregate these predictions and determine robust solutions through what is called the wisdom of crowds. So let me tell you a little bit about the wisdom of crowds, which is a fantastic thing that has mathematical underpinnings. But one of the typical stories that it illustrates the wisdom of crowds comes from a famous statistician, one of the founders of modern statistics, called Francis Galton, who in a paper in 1907 called Vox Populi wanted to see, in his words, the trustworthiness and peculiarities of popular judgments, especially when it comes to elections. He was a Victorian aristocrat, and he didn't believe in democracy. He wanted to prove that, though. And so what he did was he got data. He was a statistician, and he liked data, and he got data from a county fair where they were asking people to play a game of estimating the weight of this ox, of an ox. And the different people would submit a card with their predicted weight, 1987 pounds, 1,278 pounds, and so on and so forth. And so one of them, the one who's closest, would win, right? But Galton said, let's do some statistics of this. And he took all these cards to his house, and he computed the mean and the median and the distribution of predictions. There were about 800 predictions. And he found that the mean of all the predictions was 1,197 pounds. And when the actual weight of the ox was 1,198 pounds, which comes to say, as he concluded at the end of his Nature paper, he says, the result is, I think, more creditable to the trustworthiness of a democratic judgment than might have been expected. So he concludes that there may be wisdom in the crowd, some collective intelligence. And we have seen this over and over. I will discuss a little bit of this later on. But this is one thing that we can do when we have many algorithms, not just one that tries to solve the problem, even when some overestimate and some underestimate the actual object to predict. Now, there are many platforms and organizations that do challenges. I will focus on the DREAM challenge, but there are others, like we just referred to CASP. Some of you may know Kaggle, which is a machine learning heavy, everybody likes to win in a DREAM challenge. There are some that are, opportunistic for specific problems, like the Clarity Challenge. And there are some that are more about not benchmarking necessarily, but let's try to solve a problem of big data, like this one, CAMDA, critical assessment of what used to be called microarray data analysis, and now is massive data analysis, because microarrays are no longer fashionable. We created DREAM in 2006 out of a need to benchmark algorithms for biological network inference. And since then, we have been growing a community, organizing challenges, conferences, and trying to get people to talk to each other. And you can go and check our website, dreamchallenges.org, where you will see all the challenges that we have made, all the papers that we have published, and so on. Our mission is not just organize challenges. We are not just a platform. What we like to do is to actually solve scientific problems using the wisdom of crowds, using collaboration. So our mission is to contribute to the solution of important biomedical problems, and to do so, fostering collaboration between research groups, because we think that collaborating is a way to overcome some barriers, offering data when data is not necessarily easily available, accelerating the research, as we said before, by multiplexing kind of the groups that are trying to solve a particular problem, and assess algorithm performance, of course, is at the center of our mission. So let me, maybe for some of you this is going to be obvious, but let me, just to make sure that we understand, talk about the structure of a DREAM challenge. So typically, there is a problem, and typically we have a ground truth. We will discuss more about whether ground truths are really gold standards or copper standards in a little bit. But suppose that we have a ground truth. For example, suppose that we have a group of people that have rheumatoid arthritis, as we did one challenge, and we want to see who of these people respond and who of these people will not respond to anti-inflammatories of a particular sort. So the question is, will a rheumatoid arthritis patient respond to a particular type of anti-TNF-alpha anti-inflammatories? Now, what do we know about them? We know who responds and who doesn't respond, but also we know clinical information, we know genetic information, and we know demographic information. So we will give that data, demographic, genetic, and clinical information, to the world who wants to solve the problem. And we give a training set from where they can make the association of what of these covariates best represents or has the best information about who responds and who doesn't. And then we crowdsource this data, and people will try to solve during a period that could range from 3 months to a year, depends on the challenge, and they will eventually send a prediction. And the prediction is John Doe responds, Jane Doe doesn't respond. And we can say, because we know whether Jane or John Doe responded, so we can start to compare the degree of response that they predict, and we make an objective evaluation. At the same time, we have given this data probably wouldn't have been given in this way with a very concrete question. And we have tested a lot of algorithms to do that, and eventually these people will collaborate. So this is basically what we are trying to do. Of course, in reality, if it is a translational challenge, we usually don't know whether a patient responds or not, or which genetic mutation this patient has. So what we do is use the best algorithm or aggregate of the best algorithms to determine whether a patient will respond or not. And this is where this is important, is to use the best methods now, card-carrying best methods through a blind challenge to determine future outcomes in patients. The typical way in which we organize these challenges starts with a scientific question. So you have to have, and we will try to discuss 3 challenges and a few ideas for extracellular RNA challenges. You start with a scientific question that seems to be hard to answer. And you assemble a team of people like for example, a coordinating center, and we work together with them to try to see how to best organize the challenge that answers the question as best as possible. We have to make sure that we have the data with the right approvals, IRBs, data user agreements. When we crowdsource, we typically do a dry run to see whether we can solve it to some degree. We don't want to be the best at solving it. We don't have the time, but typically we try to see whether it's so impossibly different that all that we do is random, or is so trivially easy that, you know, we solve it no problem, in which case we don't want to crowdsource this. But if the problems seem to be good, we put them in the web in ways that we will show, you know, how to store it in the cloud and do what we call model to data solutions. We think of incentives and we start to advertise and communicate the existence of the challenge. Eventually, the challenge goes to an open phase where people agree to the rules and submit their analysis. We score them, we determine who's best, who's not best, and eventually we go to a community phase where we together write a paper trying to get to really ask ourselves, what did we learn? You know, how we learn something interesting out of this challenge. Was it some black box thing, in which case we don't like it, or is something that we really learn about the question, about, for example, if it is the I will show later, predict isoforms in a transcriptome. Have we learned the reasons why some teams predict well and some teams predict bad, and so on and so forth. And eventually we archive it or can go into a continuous benchmarking for future benchmarking, not in the context of a competition. And so these are a few of the we have run about 65 challenges so far, and all the challenges have been published. Well, not all, but a great majority of the challenges have been published in reasonably good journals. One, for example, we published in Science, in Lancet, in JAMA, in Nature Biotech, and so on and so forth. The reason is because, and this is one of the incentives, right? People submitting to the challenge know that they may have a chance at a very good journal, publication at a good journal, which for method developers is not a given, right? When you do a method paper, usually you publish in more specialized journals. And but the reason why we can publish this first is because it's our way to make sure that we learn something from the challenges. But the second is we need to incentivize people to participate, and this is typically a good incentive. So we have about 25,000 registered participants, run about 65 or more challenges, published in many papers, some ours, some people using our data, publishing their papers and using the benchmarks and other algorithms that are in our benchmarks. If you look at our papers, we have an H-index of 42 in aggregate DREAM and about 9,500 citations. This goes to the impact of the effort, right? It's not completely ignored. Many people have used the data, DREAM data and DREAM challenge data to write their thesis. I counted 20, but I stopped counting a number of years ago. We have organized conferences because we like to always make sure that what we learn is communicated, right? That if a team finalizes and wins, they share their method and why they won. And not only that, it is a requirement that people share their methods with an open source license and deposit their code in our websites. And so on and so forth. Okay, let's discuss now a few challenges and start by thinking a little bit about ground truths. Gold standards would be great. Sometimes you don't have the perfect gold standard. It has to be silver or copper standard. But there is this nice paper by Vivian Marks, technology writer for Nature Methods, in which she describes a lot of the ups and downs of benchmarking with genomic biomarkers. And some of the things that we will discuss in a second is benchmarking requires a ground truth to compare predictions with, but we not always have a clear gold standard. In some cases, people use synthetic data, and that could or could not work. We have seen that it works when there is nothing else that can do the job. It's not just a mathematical or computational exercise, it's that we need to understand at least a poor man's understanding of how well an algorithm works, even if the data is synthetic. We will show something in a second. And, but you can also imagine that you can finalize the challenge, see the predictions, and then do experiments to verify the predictions. That we have done as well. Let me start with one challenge that was a synthetic, in which we use synthetic data. DREAM started mostly as an effort to benchmark network inference algorithms. And we don't know the networks in the cell. I mean, typically these are cellular networks, and we don't know the transcriptomic networks, we don't know the proteomics networks. A lot of effort has to go in order to understand how the transcriptome is regulated, as you surely know. So we did a challenge to try to see whether we could unearth interesting results from it. The challenge consisted of 4 data sets. One was an in silico data set. That means that we imagine ways in which a network is regulated, created the transcriptome based on some differential equation ways of control. And then once we have the transcriptome, we can put them in the form of arrays, and we can simulate different conditions. So this was completely a simulation. We also had E. coli data, for which we have 805 arrays and 487 conditions, and we copied the in silico to the E. coli numbers, but the E. coli was real data. We had some yeast data, and we had some Staphylococcus aureus data. Now, this gold standard, we knew. It was in silico. We created the network. We created the simulations. This one, we knew just from the known biology of E. coli. And we know it to some extent, but there probably are connections that we are not seeing. So there might be false positives that are not really false positives. That means someone says this gene controls this other gene, and maybe we say no, it's not known, but actually that happens under the right conditions, and we didn't have it in our gold standard. For yeast, it was even more difficult because less data there was less data for yeast than for E. coli. And for Staphylococcus aureus, we only had the data. There was nothing. We created a prediction out of this. But let me tell you what happened in this challenge. So there were 29 methods that were applied, and there were 6 off-the-shelf methods that were kind of popular in those days. Some of them still are. And so we applied the methods. Well, we didn't apply. We gave the data. They had to predict the network, and we compared the network that was predicted with the network that we had. So here are the results. And the result says that no one size fits all. So let me be more clear about what I mean for that. If you want to cluster in a way the data into regression, not the data, but the algorithms into regression-based algorithms, of mutual information-based algorithms, or Bayesian networks-based algorithms, or random forest-based algorithms, and so on. In each of these categories, for example, the one that was best so the one that was, let's see what I want to say. This guy Meta was best in the yeast barely, right? This line, dashed line is random. But this was not the best in E. coli. Some other algorithm was best in E. coli. And that was not the best in the in silico. This was the best in silico. So there was no one algorithm that fits all the network, because there are peculiarities in our synthetic data, there are peculiarities in prokaryotes, in their regulation, there are peculiarities in eukaryotes. So we had a mix of all. So clearly, it's not just give me the transcriptional data and I will do the work of predicting the network. So if there is no one size fits all, can we aggregate in a way that Galton did with the weight of the ox and create a community prediction that somehow is the aggregate of them all? And it turns out that if you do that in some score that has to do with the multiplication of all the p-values, the community is the best overall. And it makes sense. If none of them is good always, then the aggregate of them will have the best of them all. But there may, and furthermore, and this is an interesting thing, is this is the overall score of reconstruction of networks. This is the variance of individual methods, of the 29 individual methods. Now, if you take 5 at random and aggregate them, then the overall score has also a dispersion, but the median goes growing as the number of methods that we integrate increases. This variability has to do with that the 10 methods are taken out of 29 possible methods, and so there is a variability, but always you see that more methods will have better score than just one in terms of the distribution. So this is one of the things that I want to leave you with, is that there is a way to aggregate data that will not degrade with the bad predictions, but will be enhanced and be more robust because of the good predictions. And this, by the way, is a prediction of the network of Staph aureus, that now we can do the wisdom of crowds of all the predictions Staph aureus, and some of these connections can then go and be tested in the lab. We didn't do that, but that was the idea. The idea is that now we have benchmarked a number of algorithms for network inference. Let's see which are the best network in Staph aureus based on the aggregate. And now we have a prediction that we can go and try. Some of these were tested, not by us, with good results. Okay, the previous model was the data went to the modeler, but in order for this to happen, the data should not be public. You give the data, you don't give the ground truth. But the people who created the data should be willing to give the data, and that is not always the case. We didn't ask models to come back. We asked simply for predictions. So the people get their data, the validation data without the labels, and then they have to make predictions, and we compare with the ground truth. The only thing that we have is the predictions. We don't have the models. And that's a problem because we cannot enforce model reproducibility. And there are other problems with this way of doing things, though it's a good one, and typically is the one that Kaggle uses in the very popular competition platform. But we are proposing a model to data platform. We have done many challenges using this platform. It's basically what happens is that people train data, maybe with public data sets, maybe with private data sets, and they produce a model that they put in a container, so to speak, which is called a Docker container, for the ones who are more familiar with this technology, and they submit their model for us to run it against a hidden validation set. and that allows us to score it, but the validation set, which is precious because probably hasn't been published or has commercial value, is never downloaded or seen by the challenge participants. However, we can benchmark their algorithms, and this is the model to data paradigm. Let me skip the digital mammography challenge discussion and go directly to another that is a little bit more relevant to the problem of extracellular RNA analysis, which is what we call the somatic mutations calling RNA challenge. It's not really looking for somatic mutations, but this came after a series of challenges in which we were trying to look for somatic mutations in cancer patients, for which a pipeline is not at all clear. You don't know in a cancer patient which really are their mutations, and if you run two algorithms, you might get two different results. But in this case, the question was different. We wanted to ask, can we predict or detect RNA fusion, and can we detect, this would be RNA fusion detection through RNA, but the fusion is of DNA, and can we detect the different isoforms that the different splice variants from the RNA-seq data. This paper, I think, is going to be eventually published in Cell Systems, but we don't have the final yes. And the paper was mostly written on the challenge carried on by Kyle Elrod from OHSU. The question is, can we detect alterations in the expression profiles of known RNA transcripts, known by us but not by the participants? And can we detect fusions between different parts of the genome? These are all the people who participated as sponsors of this challenge. But here is the important thing. We had, our input was synthetic data simulated by using an RNA-seq simulator, trying to reproduce all the quirks of non-RNA-seq, and spiking data in 5 different cell lines. And so we spiked in fusions, we spiked in isoforms. And so once the data was then sequenced or the synthetic data was produced, then we also had the fusion truth and the isoform truth. That we will use to compare the predictions that different methods did based on the data, but not knowing the ground truth. And then eventually we have scored against our ground truth using different evaluation metrics such as the correlation, Kendall, beta correlation, and sensitivity and precision. Okay. The challenge started in July 2016, finished about a year later, and the paper is being finalized as we talk. There was a round of post-challenge community phase, and they tried to use long sequence Oxford nanopore sequencing in order to detect transcript isoforms, but unfortunately that didn't give us enough coverage and we couldn't use that. So we had to use another way to test. But the first 1, 2, 3, 4 rounds were tested on synthetic data. And here is where they learn about how the algorithms is going through a leaderboard. But the final results were tested on spiking panels, not on simulated data. So just to have a sense, each of the 5 cell lines had 5 replicates, and the 5 replicates had 20 transcripts isoforms from 5 genes with 3 to 5 transcripts per gene, and the fusions had 40 synthetic fusions that were spiked in the cell lines. And for each of the transcripts, we spiked with different concentrations that went from 0, actually there was a control with no spikes, 5 picograms, 25 picograms, and 50 picograms per milliliter. Now, the first surprise, and I want to be finishing in about 4 or 5 minutes, is that the isoform challenge, the y-axis here, by the way, is a measure of correlation or concordance index called the tau beta statistic. So 1 is perfect. I think 0.5 is random. What happens here is that none of the participants was better than a baseline method that we used. So we used typically a baseline method. If there is a method, we want to see that like AlphaFold was better than the 2016 CASP, right? So we want to use something as a baseline to see whether people are doing better than the state of the art. And actually, nobody did better than the state of the art. These are the p-values of the comparison with the organizers. And so here is an interesting conclusion from this challenge, right? That it seems that we have plateaued in our, at least, you know, if you take this challenge at face value, we have plateaued in our ability to detect isoforms. The existing methods do not differ from each other and do not differ from the one that we used as organizers as a baseline. I think this is a negative result, but it's an important result to have into account. For the fusion, something different happened. Now, in principle, we have the simulated data and we have the spiked-in data, but the spiked-in data had a problem. The problem is that the algorithms could call a fusion, and there could be more to the call fusions than the spiked fusions. For example, these fusions were spiked, 40 of them were spiked in cell lines that already had their own fusions because they are cancer cell lines that are pretty screwed up genomes. And so the background fusions could be detected by the algorithms, but if we only use the spike fusions as ground truth of positives, then we will call anything that they discover that is true but is not in our ground truth as a false positive, and that would be unfair. And so what we did was, that was a known unknown. We didn't know which ones they were, the baseline fusions, but we know that they were there. So what we did is we used the same predictions and we detected how many of them, of the predictions, coincided in calling something a fusion. And we added that to the ground truth. That was what we call the imputed ground truth. And imputed ground truth, well, there was a procedure for this that is in the paper. I can discuss this more. In general, was validated with external data sets, for example, the CCLE Cancer Cell Line Encyclopedia had a number of fusions that they report, and most of the fusions that were predicted by the majority of the algorithms submitted were also part of the Cancer Cell Line Encyclopedia, not all. And when we looked at it, it's about 90% of our imputed ground truth had additional evidence that those were real fusions. In any case, there were two clear winners, and you can see the values of the F1 score, which is some sort of average between precision and sensitivity, the harmonic mean of the precision and the sensitivity. This participant, Sebastian Jurek, from the German Cancer Research Center created a method called ARIBA that was pretty good. The F1 score was 0.74. The second best was Brian Haas from the Broad Institute that had 0.7 and 0.63. And the next ones, this was our baseline, 0.59 was sufficiently lower than the best that we can say that they are really discovering something that we didn't expect from our baseline models. So this was, and in the paper we discuss what made these algorithms be better than ours, and typically the short answer is pre-processing. Very good. So let me finish with this slide. There is more later, but we can discuss if there is time and the appetite for it. The question is, can we use a dream challenge for extracellular RNA analysis? And the problems where benchmarking seems to be necessary are the following. In my view, you guys probably know much better than I do. One topic is experimental protocols. For example, extracellular RNA extraction protocols from carriers, whether first you have to separate exosomes using size exclusion or ultracentrifugation, or vesicles or lipoproteins using different ways, and different methods will give you different results. So there is one problem with benchmarking, which one of these is best. Second thing is sequencing short reads. We all know that RNA extraction is not straightforward. Different ways of extracting will give you different results. The library preparations have their own problems. The sequencing itself, can differ depending on which platform used to sequence. Now, at the computational level, these short RNA-seq have to be mapped, and that could be complicated. And a comparison of methods to do that might be useful. Also, thinking of an aggregation of different methods in order to tap on the wisdom of crowds. And finally, there are translational challenges, such as biomarkers based on extracellular RNA, be it for disease, the ones that are indicative of which fluids you are extracting the extracellular RNA from, the tissue of origin of extracellular RNAs that you are interested in using as base marking. Very good. So I have a few more things about the concept of challenges, but let's see whether this picks up in the conversation later. My conclusions are the following. Benchmarking competitions contribute to transparent and reproducible, I say AI research, I should say data science research and computational biology. The extracellular RNA community, I think, needs to find the right problems and the right data to benchmark some of the algorithms in order to make sure that the results are reproducible and verifiable. Finding a ground truth that is good, even if it is a copper standard, if it is not the perfect ground truth, is good for the field. Even doing synthetic data benchmarking, I think it would be good for the field, or spiked in. The data, if anybody is willing to provide data for this effort, will be protected through a model to data paradigm. That means the data will, don't worry about being scooped. If you produce the data, you will publish first and people will not be able to have it. The only thing that they will have is how well the algorithm fares against that data. And again, when many algorithms are at hand, we can tap on the wisdom of crowds. And if we do a challenge, we will have many algorithms at hand for different problems. And this could be a good strategy that will make the results more robust and more generalizable. This is for different challenges, there is different teams, but this is the core team of DREAM, what we call the DREAM directors. We have been discussing these particular challenges with Julio. Many of you know him, but Justin Guiney is now the DREAM chair. When I stepped out, earlier last year, Paul Boutros from UCLA, Julio Saez Rodriguez, Jim Costello, Laura Heiser, Pablo Meyer, and Julie Bletz. Thank you.
Thanks, Gustavo. Let's jump into questions since time is short. Joel Rosowski asks, for the Docker version of submitted algorithms, are there restrictions on or limitations on runtime on the submitted algorithms? Or
it depends on the size of the cloud. So if I understand the question is, if you submit a Docker, if it runs for 5 days, will we stop it? And the answer is yes, most likely. But typically what we do is we have a fast lane where we test the algorithms for sanity with respect to a short amount, in a small data set. When it works well in that data set, that means that it doesn't have obvious bugs in read in, read out, and computation, then and using the cloud, then we move it to the actual lane where it taps on the actual data. And we can, and it depends on the amount of cloud that we can get, right? If we have 100 GPUs or processors, then we can be more lenient. If we have 20, then we have to be more stringent and allow everybody to participate.
I guess I would say that what we as a community need to do now is really think about how to, as Gustavo said, find the data and find the question to ask. I think we have a good challenge in the biomarker discovery area. I was just thinking just now, based on conversations with some people, would it be possible to make a challenge for improving the baseline annotations of some of the classes of non-coding RNAs? Gustavo, do you have any thoughts on that?
Yes, I think that that is definitely a possible challenge for which, again, part of the puzzle is to find the right gold standard, right? So how would you do that?
And a question I guess I would direct to Joel. Again, we're sort of in the discovery phase. We're trying to brainstorm. Referring sort of to your question about computational requirements, if we tried to create a challenge for, finding the best short RNA-seq method, short RNA-seq read method, and ran excerpt against various competitors, what kind of, that seems computationally intensive and might be a difficult thing to put together. Do you have any thoughts?
Well, actually, I've got a thought and you can hear me?
Yeah, we can hear you.
Oh, cool. Just wanted to make sure. I mean, that would be interesting. I mean, I guess related to your question, which maybe Gustavo could answer, in terms of calibrating or comparing a sort of algorithms, do you have a thought where the sort of the prediction is something that's sort of just quantitative, like the level of expression or the abundance of something versus sort of presence absence calls? Do you think that makes a difference in terms of comparison?
So it depends on the. So, for example, you can have a very good score when it comes to classification, but not so good a score when it comes to correlation, for example. So it depends on the. So if you compare abundances versus compare presence or absence, right? Classifying detected or not detected versus classifying how much detected it is. You will use different metrics, and but usually what we do when we are unclear about what is the right question is to have two sub-challenges. One could be, predict presence or absence, and the other one is predict abundance. And then we answer the question empirically.
Yeah, no, I think that's important to have that sort of distinction, because like the example with your fusion transcripts, the example you gave, I mean, the actual exact criteria is really important. Are you trying to predict the fusion transcripts? Is there a requirement that it has the exact coordinates, nucleotide, of the breakpoints and everything, or is it just rough, prediction of where it is sufficient?
Exactly. So all these things are important. In principle, the fusion has to be predicted at the right breakpoint. But when we were looking at whether the ones that were predicted existed in other data sets as fusions, the breakpoints where they were predicted were different. So all these things can be evaluated as well, right? But in principle, yes, the metric has to be to answer the question that you need to answer, right? And if what is important is exactly where the breakpoint is, then you have to add it as an example. The same thing with isoform challenge. If you want just to see the exon usage, or you want to see the abundance of the whole transcript, right? These are different questions that should be answered differently, but depends on the gold standard that you had.
We're over the hour. Let me just end with, I think, a good comment from Brici Rascom's-Heater, and Gustavo can comment, and then we'll end. And say that I think that benchmarking and comparing different normalization techniques for exRNA analysis is necessary as well, because of the much lower RNA complexity and less RNA in biofluids compared to bulk tissue.
Yes, absolutely. I think that would be a great challenge. I think that
can you think of a way to create such a challenge, sort of off the top of your head, that solving the main issues, find a copper standard sort of thing?
Yes. So one could imagine the creation of a gold standard that is either with it depends. If we want to be not use any substantial amount of money, we can just do a synthetic challenge in which we create transcripts that map with different degrees of precision, with different error rates, and with different complexities. And then we will be able to assess how well different algorithms do at different complexities and matching accuracy. But so these things can be done synthetically. I think it's not so difficult from the point of view of detection, right? Because after all, you can very well imagine what are the biases in real data, and use those biases in the generation of your synthetic data set, because otherwise it will be too obvious if you are off the distribution to detect what are the synthetic reads that you have put. I don't know whether that was clear, but I think that it is doable. It is doable synthetically. You can add that to real data, right? What I'm saying is you add reads to the files where you have the raw data, and you have the FASTQ file, and then you expect them to match it to the right place, and you will see whether they do or not.
Okay, thanks, Gustavo. So this is the beginning of a discussion. It's a complex topic. We'll return to it at the April workshop. And I'll just end by saying that if you're interested in doing a webinar in March, let me know. The first Thursday in April, we're going to have Peter Mestag from Belgium presenting his work on the Human Biofluid RNA Atlas, which is basically like the European exRNA Atlas, which has just been published. So that should be very interesting. And thanks everybody for participating.
Thank you. And if you have any feedback, please shoot it away.
Thank you.
Great.